There was a great already mention of this data by Caitlin. So, Conrad.
Awesome, thank you. Yeah, so thanks everyone, and thanks for the organizers for giving me the opportunity to speak here. I'd like to convince you that, I probably don't need to convince a lot of people in this room, but that rare variants are really important, and that they present, but one interesting component is that they present new opportunities to identify causal mechanisms for diseases, not just rare diseases, but also complex traits. So we've already done plenty of GWASs. We've found thousands, hundreds of thousands of robust loci for complex diseases. And of course, on the other end, Mendelian disease studies have given us true insight into high-impact, ultra-rare variants for rare disease. But there is this kind of intermediate in between where you have these, sections of intermediate frequency variants that probably contribute to diseases as well. And so in this figure that I'm showing on the right is from analysis in schizophrenia, in which you can see plenty of variants that have been identified by GWAS here in the bottom right, so fairly common variants and very low effect sizes, as well as some more kind of high impact variants, almost getting close to de novo level variants that really have really strong effect sizes and genes for that matter. So especially copy number variants fall into this category. But you'll notice this middle section here is not really filled in, and some of that is there's a little bit of a challenge in terms of a little bit of sample size, a little bit of natural selection that kind of tamps the frequency of these variants down, and identifying the variants in this section really requires a lot of sequencing, and so. But there may be opportunities to use these kind of data as they grow to find insights into common disease. So of course, as everyone well knows, these biobanks enable what we call these all-by-all analyses. So looking at millions of genetic variants or lots, burden tests of each gene, and looking across thousands of phenotypes. So being able to understand whether an individual variant has an association with a trait, or whether variants collectively in a given gene have an association with a trait. The UK Biobank is one such example, as already mentioned, a large collection of about 500,000 individuals with matched genotypic and phenotypic data. But of course, one thing that you might notice from this, or you might already be aware of this data set, is that it has, as has been pointed out in many cases, a very Eurocentric bias. The vast majority of participants are of European genetic ancestry. And we've done some analyses where in this Pan-UKB project in collaboration with Alicia Martin and others, where we can actually see that even adding the small fraction of individuals that are non-European genetic ancestry adds a lot of information as to genetic discovery. So even though it's a very small fraction, it actually adds an outsized proportion of the data, and that data is already there. It's not new data that needs to be collected. So, one thing I would do is encourage everyone to look at all the data in your data set, not just the largest ancestry group, because that really gives a lot of increased power. And so, the other end of the coin for UK Biobank is what we've been able to do is also do this kind of rare variant association analysis with the exomes and now the genomes. I'm sure others are doing. We've released this as a browser in GeneBAS, where you can kind of browse your own favorite genetic variant or gene and see whether there's an association with rare variants in these genes. But so these, like I already mentioned, this resource is very comprehensive, but lacks quite a bit of diversity. And there have been other resources, in particular, the All of Us cohort, as already mentioned, that increases quite a bit in terms of the scale of diversity. So this is a cohort sequenced within the US across many sequencing centers and recruitment centers. And it really does have an unprecedented amount of diversity. Over 80% of the individuals are individuals who are considered underrepresented in biomedical research. And close to half of them are as such because of their race or ethnicity. This cohort has about coming up on 500,000 individuals ready for sequencing, but what data I'm going to describe today is from a call set from this past year, where they've had about 250,000 individuals, 245,000. I believe there's a new release planned for later this year with a little over 400,000, and we'll update these slides when we do those analyses. But for now, I'll describe the kind of preliminary work on the first data set. And so here you can see the number of individuals by genetic ancestry group. Again, still the majority is European ancestry individuals, but with a much smaller kind of size of that majority, and the number of variants in each ancestry group. So we've built multiple pipelines in particular to kind of pull together a lot of these data, in particular the phenotype data. So this is in collaboration with Robert Carroll and others at Vanderbilt, where they've kind of done a lot of the curation and designation of the fee codes and various kind of electronic health records, as well as pulling together data from the genetic information. So Wenhan Liu in my group has been spearheading a lot of this effort, and in fact a lot of the effort that I'll talk about today. When we pull these together, we get about 3,400 phenotypes across these different categories with at least 200 cases or measurements per group. So for the quantitative traits, including these lab values and physical measurements, we kind of consider most of them. And then for binary traits, we look at all these diseases that have at least 200 cases. As well as, so we've looked at both the fee code data as well as the personal family health history surveys and medication use as well. When we look across these data sets, so if we look at UK Biobank against all of us, we do see that phenotype prevalences are generally concordant between the two, suggesting that these are finding similar signals of complex traits. And one critical component to this analysis was that it turns out when you do these kind of analyses, especially when you have these very diverse cohorts, it might seem attractive to put everyone together into one model and run one big association test. However, when you do that, you get a lot of population stratification. This is something we've known about for some time, but even with these kind of novel mixed model methods, you still see this increased stratification. And so what we've done so far is to split each of these, this cohort into 6 genetic ancestry groups. But even then, even when we do this kind of split, as we've done in many times before in Nomad or in UK Biobank, you still see a lot of residual stratification. So there's actually another step after that of kind of sample pruning to figure out what is a slightly more genetically homogeneous set that will not show stratification. And when you do that, you actually get really good results. So those are the ones I'll talk about today. As I mentioned, we've built several of these pipelines, and now I'm showing the kind of large pipeline to pull together the phenotype data and the genetic data, and to run, we've run mixed models using SAGE and SAGE-Gene. One critical component to understanding whether our tests are working, and in fact, this previous mention of stratification was why we really realized that we had to do this, was what we've done is we've actually just taken phenotypes and taken these individuals and randomly generated phenotypes for them, heritable traits using just the genetic components. And when we do that with the data set that we have, we actually see very good calibration for our summer statistics. Before we did the kind of sample pruning, you get really wild QQ plots. And so this kind of gives us really good confidence that our data look pretty good for quantitative traits and for common binary outcomes like 10 to 20 to 50%. For more lower rarer outcomes, so less than 1% or so, you do see a deflation indicating that we're underpowered for these, especially for the smaller ancestry groups, which is pretty expected. As I said, we've run, so at this point, these 3,400 phenotypes amounted all in all, when you split it by genetic ancestry group, to about 8,000 GWASs and rare variant studies. We've now run this this sum total of this was about 500 million association tests, sorry, 500 billion association tests, I forget where that is. But we managed to parallelize this to a point where we've implemented it in this new Hail batch system that scales these association pipelines and allows us to complete this pipeline in one weekend, even though it was about 700,000 CPU hours. These data are really, we've only started to scratch the surface on digging into the data, and we really see a lot of great signals that we kind of might have expected, including some validation of a recent observation from the UK Biobank of rare loss of function variants in GIG-YF1 for type 2 diabetes, published by our friends at Alnylam in 2021. And as well, finding really interesting signals that may not be necessarily quite as well known. So for here, I'm showing a Manhattan plot of triglycerides. This is a rare variant Manhattan plot, so we do not expect the kind of peakiness that you would normally see. Because each of these points is a gene, not a kind of region, or a SNP for that matter. And so we see a lot of great associations that we've well known for a long time, but we find this new one, TIMD4, which does have a GWAS hit. It's not that new in the sense that we've known that there's something interesting at this gene, but the really interesting thing, we can actually see that rare loss of function variants are driving increased triglyceride levels. So really getting to much more of a functional point than a kind of traditional GWAS that just says there's something interesting here. When we compare the UK Biobank to all of us, we do see kind of concordant effect sizes as expected for these rare variants, for the rare variant tests, which is great because, like I said, the phenotypes are similar and the, sequencing was high quality, so really getting to nice, data QC. We've built yet another pipeline for summary statistics quality control where we've really kind of had to dig in and understand, are there any phenotypes that fail QC? You really get, when you do 3,400 phenotypes, usually a few slip in that you have to get rid of. And so we've done that, and then finally the last step is doing meta-analysis between the gene burden analysis results. So again, I mentioned we've done about 6 genetic ancestry groups, and we've kind of combined them using a actually very classical method from the 40s, Stouffer's method, that considers the direction of effect as well as the significance. So in this case, because these loss of function variants, you know that they're doing the same thing to the gene. They're ablating the expression of the gene, or at least predicted to. They should all have the same effect. And so when you put them together in a meta-analysis, it's even more powered if you considered the sign of the beta as well. So we've done now, like I said, thousands of phenotypes, and so we can show these little mini Manhattan plots. So each of these sections of the plot is a little mini Manhattan plot, and there's lots of associations, many well-known associations. The vast majority of the ones that near the top are well-known, but we're starting to dig in if there's anything interesting in these particular kind of sections down here. And so, for instance, there's a few that I can share that we've recently discovered that are, again, many of these are well known in the literature, but not found in UK Biobank. So really harnessing a second data set suggests that there's more power and more discovery to be had. In particular, MLH1 loss of function variants with colon cancer, and this one that has recently been reported in a large scale hypothyroidism scan in P10 loss of function variants. But again, also getting at not just the GWAS, not just the association locus, but the actual direction of effect, so showing that these loss of function variants increase or decrease disease risk. We released these data this past week. You can now actually access these on the All of Us Researcher Workbench, which is now available to the international community as well, international academic community, I should say. So there's now some user support hubs that you can, pages that you can see. We've actually put out a lot of information about how you can access the data, and pretty soon we'll have a featured workspace on the All of Us Researcher Workbench where you can actually kind of, with some helper functions that you can use to access the data a little bit easier and better. But for now, you can access the data now in full matrix form. So these are something like 22 terabytes of data, totaling about 70 terabytes of data you can access right now. And also per phenotype. So if you have one phenotype of interest, you can access that a little bit easier than having to trawl through 70 terabytes of data. And so finally, because again, still even these kind of analytic frameworks are quite challenging, and they require sophistication in Python and these Hail frameworks that we've built to do this. We've also been on top of this building a new browser framework for multi-ancestry, GWAS, and RVAS. So the GeneVAS project that I mentioned previously with UK Biobank, we've built this browser where you can kind of look at your favorite gene or trait or variant of interest and look at the Manhattan plots or QQ plots and kind of see what associations might pop up. We've been building the same thing for all of us, but actually in a much more larger scale. So in this case, it's both using rare variants and common variants. Overlaying those two is a little bit of a challenge we've been thinking about, but haven't really gotten a great way to do yet. And on top of that, looking at views from multiple ancestries. So most of the time, you want the most powered one, you want the meta-analysis, but being able to see the component ancestries to figure out which ancestries are driving the signal. Are they concordant? Are they not? Is a really interesting thing as well. We've been making some progress, in particular at the scaling components, because serving 70 terabytes of data is a bit of a challenge. Usually when people try to go to a website and they have to wait for about 2 or 3 seconds, they usually give up. So showing things at 100 millisecond speeds is a little bit of a challenge. But we've been making some progress in downsampling and caching to kind of show this quickly, and some of these visualizations for these kind of what we call Miami plots to show both meta-analysis, for instance, and per ancestry groups. So with that, I'd like to thank everyone involved in this work, especially Wenhan Liu, who spearheaded a lot of this effort, but a lot of the folks at the All of Us cohort, the All of Us program, as well as the cohort itself, the participants. And with that, happy to take any questions.
So rapidly, we have time for one question from Conrad. And please always remember to state your name and institution.
Okay, I got one question. My name is Martin Kellerman. I'm from University of Cambridge. You mentioned that the mixed models couldn't fix the stratification issue. Could you tell us a little bit more about that, how that happened, or what was the problem in?
Sorry, it's hard to hear up here. The mixed model had a...
Yeah, the stratification issue was not fixed by them.
Yeah, so just to be clear, it's not that the mixed model had it, but the linear model didn't. The mixed model does improve the stratification because you add the genetic relatedness matrix as the random component. But the problem being when you look in the PC space and you kind of draw, we use a random forest for ancestry assignment, and these random forests have tended to be a little bit more permissive in the past, just to catch a little bit more individual people. But even though, in this case, you still see quite a bit of smeariness in the PCA plot, I guess is the right word to use. And so when you kind of look at that, when you include all individuals from that, you still get stratification. And we see these stratifications when we do simulated phenotypes. So what we do is we actually just use the genetic relatedness matrix to simulate a heritable phenotype and then run a GWAS of that. Yeah. And in many cases, the mixed models step one, which is this null model, doesn't even converge because it sees this phenotype that should be heritable, and yet it's heritable in a kind of unstable way, if that makes sense. I'm happy to talk about it a little bit more offline if you'd like, but that's the long and short.
Thank you.
Yep. Thank you, Conrad.
And then we'll move to the third speaker of this session.